So with the continuation of pathogenesis of cancer, we're going to start what we learned already, and then we're going to correlate it with the pathogenesis of cancer that we're going to learn from now on. So from here on, we have this ligand and the receptor functions. How do ligands attach to the receptors and which ligands attach to which receptors, and how do we differentiate them on the basis of their mechanism of action? Let's start with, first of all, tyrosine kinase. Most of all, you'll see it written in like RTK. It's actually tyrosine kinase pathway. So what happens in tyrosine kinase pathway? Let me give you a brief overview of tyrosine kinase pathway. So tyrosine is actually an amino acid. It's a bunch of amino acid. That's what tyrosine is. So tyrosine is present on the membrane of the cell, and it's present here and it's embedded in that cell membrane. It has its upper end, it has its lower end. On the lower end, which is intracellular, it has kinase molecules attached to its base. Those kinase molecules may be 1, 2, or 3. And it's never possible that there's only one tyrosine molecule present. It's always going to be in pair. They will be far away from each other. They will be distant from each other. And whenever any kind of ligand comes and binds at these receptors, these tyrosine kinase receptors, what happens? Those kinase molecules, they're going to get phosphorylated. How are they going to get phosphorylated? ATP is present inside the cell. Whenever a ligand comes in and binds at the tyrosine receptor, a reaction comes through and kinase phosphorylates themselves by the help of ATP and converts the ATP into ADP. Slowly, all of the kinase molecules are phosphorylated on both ends, not on one end. It's not like one tyrosine molecule and one kinase molecules are being phosphorylated. It's about all of the molecules. I told you it's going to be a couple, so both tyrosine molecules are attached to a ligand, and then their kinase part, their kinase chain is phosphorylated right now. And it's getting phosphorylated with every move. So when they're properly phosphorylated and they have consumed the ATP which was required and converted the ATP into ADP, now those molecules will come together and they'll make a bond like this. Whenever this happens, this leads to intracellular messages that ligand is there to send. Once it reaches the nuclear level, it gets its desired effects, which depends on which ligand was bonded to the tyrosine receptor. So which of these molecules do you think get attached? Which of these ligands get attached to tyrosine kinase receptor? Or you can say which of the ligands use the tyrosine kinase pathway? First of all, it's EGF. Next on, EGF stands for epidermal growth factor. You must have heard of it. Then we have IGF-1, insulin-like growth factor 1. And then we have insulin itself. So these three are the examples of ligands which attach themselves to the receptor site of tyrosine kinase. So all of these attach themselves at the receptor site of tyrosine. And when tyrosine gets attached to them, it gets its desired effect by activating the kinases, and then kinases get phosphorylated, and then the whole chain starts afterwards. Next on, we have G protein coupled receptor. Before we get to know that which of the ligands use G protein coupled receptor, we have to know that G protein pathway or G protein coupled receptor are of three types. There are three types of pathways in it. First of all, we have GI, then we have GS, and then we have GQ. GS stands for synthesis of DNA. It's stimulation. Whenever GS pathway is being used, it means that any ligand who's attached to that receptor is increasing the synthesis or increasing the output out of that system. What happens if GIS system is activated? If it's using GI1, GI stands for I for inhibitory. So it will have inhibitory effect. And then comes GQ. GQ is totally different from both of these. GQ has a very unique function, and which is very important for learning the neoplasias, and we'll learn about it in detail afterwards. That GQ causes the increased calcium level, and it affects the chemokines and cytokines of the body. So, which of the ligands do you think attach to G receptor? First of all, we have prostaglandins, rentis. Rentis is actually a molecule which is very important for cell proliferation. So, if anything happens to this, it leads to increased proliferation or decreased proliferation depending upon the mutation. Next on, we have SDF1. Next on, we have extracellular matrix receptors. Now, extracellular matrix receptors mean those receptors which are present on the extracellular surfaces, extracellular sides, and then come in and get the desired effect without attaching themselves to either of these receptors or using either of these pathways. What of these ligands do use this kind of pathway? It's fibronectin, it's collagen, it's laminin. Now, all of these ligands get themselves attached to extracellular matrix receptors, and afterwards the desired effects are achieved. Next on, we have ligands and their receptors. First of all, we're going to start with nuclear receptors. We all know that steroids or any kind of androgens or estrogens, they don't need any kind of help. They don't need any kind of help passing through the membranes. They don't need any kind of help passing through cell membrane. They don't need any kind of receptor. And basically, they don't even have receptors on the cell membranes because they're permeable. They easily cross the cell membrane, get to nucleus, and attach themselves to the nuclear receptors. In some cases, by the way, there are some cytoplasmic receptors present which attach themselves to these ligands before they reach the nucleus, and then they put themselves with the ligands to the nucleus. So they can either attach themselves to the cytoplasmic receptors or they can attach to themselves to the nuclear receptors. But basically, they're going to use the nuclear receptor pathway. So they attach themselves to the nucleus, and what happens afterwards? The desired effects are achieved. The messenger RNA is produced. That messenger RNA moves up, makes proteins. So, for example, if androgens or estrogens are secreted and they reach up to that level, depends on which kind of signal we're getting from this kind of ligand or stimuli. Either it's asking it to increase the production, either it's asking it to decrease the production, either it's asking it to stunt the production. The response depends upon the stimuli. Either way, they get to the nucleus and get the desired effect by attaching themselves to the nucleus of to the receptor of the nucleus. Next on, we have serine or thiokinase kinase pathways. Now, these pathways are very important. These pathways stands for TGF beta. It's very important in order to get this kind of effect, serine and thiokinase kinase. What do they actually do inside the body? They're responsible for normal matrix of the cell. They're responsible for the normal growth of the cell. So they use this TGF beta pathway, which ensures that the growth of the cell and the maturation of the cell is normal and it's peak speed and it's getting the desired effect at the desired time. Next on, we have kinase associated receptors, which include growth hormone, T cell receptors, and interleukin 2. So what does kinase associated receptors does? I already told you the whole mechanism of tyrosine kinase pathway. So it's some kind of illustration here. You can get the point out of it. You can see these molecules are tyrosine. And at the bottom of them are attached kinase molecules. So what happens is any ligand comes in and binds at this side, and any ligand comes in and binds at this side. These bottom kinases will be phosphorylated. When they are phosphorylated, what happens is that after being phosphorylated, they'll attract themselves. They'll come closer, and then the desired effect will be produced by sending an intermediate signal, by sending an intermediate message to the nucleus to get the desired effect. And which of the ligands use this pathway? Growth hormone, T cell receptor, interleukin 2. Why does T cell receptor stay at this level? Because if T cell receptor gains entry into the cell, the effects are going to be devastating because it's going to kill the cell. Or autoimmune diseases are in the chart because it's going to be exposed to self antigens. So it attaches itself to the surface to get the desired effects. Next on we have signaling after receptor activation or action of RAS. What does RAS stands for? It's reticular activating system. Reticular activating system. Remember that we're going to use this RAS multiple times. So for feasibility, I'll use RAS, but you have to know it's reticular activating system. So what happens is the reticular activating system is normally at inactive stage. It's not needed to be active all the time. So this system is attached to a GDP all the time. When it's attached to the GDP, it's inactive. As you can see, RAS is attached to the GDP and it's inactive. For in order to get it to the active stage or active state, we have to convert this GDP into GTP. We have to phosphorylate it. When we phosphorylate it, it gets active, and then the proliferation of the cell starts, and it has some apoptotic functions, and it has some regulatory functions. It affects the growth of the cell. So all of these functions will be monitored or given, or cells will be proliferated when GDP is converted into GTP. So what happens? RAS is given that phosphate group to convert it into GTP, and now RAS is active. That GDP is gone now. That GDP is off the mark. Maybe it was GTP who gave it a phosphate group and then freed itself in the form of GDP. What gives off its phosphate group? That's the answer. GTP gives off its phosphate group, converts itself into GDP, and that phosphate group is now taken up by this GDP to be converted into GTP, so that reticular activating system might be activated, get into the active stage. So now the effects will start of reticular activating system. Now the desired effects will be met. And this goes on when the whole process, the desired effect is achieved. Now we dephosphorylate the whole group of GTP and convert it back into GDP. When we converted it back into GDP, the whole system of proliferation and new cells beginning and apoptotic features, all of that is gone now. It's in inactive stage, so no desired effects will be met afterwards. So what we have to do in order to get it activated is to phosphorylate it, and what we do in order to get it dephosphorylated is dephosphorylate the group, or in order to get it into anti-active or inactive stage, we have to dephosphorylate the phosphate group from GTP and convert it into GDP. And you can see this GDP will now get this phosphate group and get converted back into GTP, and this in cell activates the cell proliferation. Now, if mutant RAS is formed, for example, this RAS, which is an inactive form, and it's always combined with this GDP, now it has this mutation in it. Now, this was a normal functioning of RAS. Now, this was a normal function of reticular activating system. We're not studying the normal, we're studying the neoplasia, we're studying the cancers, we're studying what happens if anything goes wrong. So yes, anything can go wrong, and what if the RAS is the one to get mutated? If reticular activating system is mutated, what happens that it is always activated? What do you mean by always activated? It means it's always proliferating. It's unregulated cell growth, it's unregulated proliferation of the cells. So that will lead to the formation of cancer, the mass of cells, the tissues, which we call cancer because it's unregulated growth. And what we learned in the last lecture of carcinogenesis of the neoplasias, we learned that it's always going to be monoclonal. It's never going to be polyclonal, that these 4 or 5 cells started at this level and then they started proliferating. No, it's always going to be monoclonal. So it's always going to be very feasible for us to detect it at the site that it's either cancer cell or either it's hyperplasia of the cell. So when mutant RAS mutant RAS gene is continually active, what happens? Continuous stimulation of cell proliferation happens. I already told you, continuous proliferation of the cell will happen and it will lead to a mass, a cancer, a neoplasia. Next on, we have signal after receptor activation. Signaling after receptor activation. How does that receptor gets activated? In order to get this concept, you have to know that what P85 and what P110 genes are. These are actually tumor suppressor genes. These are the genes which are responsible for suppressing the tumors in the body if there is any. If there is any formation of cancer, these genes and many other genes that we already talked about in the last lecture, P53, retinoblastoma, and stuff like that, all of that is very important. But the topic we're talking right now about signaling after receptor activation, these two are very important. Once these two tumor suppressor genes are mutated, like P85 and P110 are mutated, what happens? With the help of P13K, RAS gets into the shape. We already talked about RAS in the last slide, reticular activating system. When RAS gets into the shape and it's mutated, because we're talking about the pathogenesis, so it's not going to be the normal RAS, that it's going to be GTP and then converted into GDP and into inactive stage. No, it's going to be active all the time. So you can imagine if reticular activating system is active all the time and the cells are proliferating and proliferating, what are going to be the effects? It's going to cause RAF. What does RAF mean? RAF means that it's going to attach itself to the receptors, which are going to increase its uptake, even increase its stimulation. Next on, it reaches the mitogen level. M stands for mitogen, extra, kinase level. It reaches the mitogen level and now it's proliferating out of its bounds. Afterward, it reaches the extracellular kinase level. At this level, it is at its maximum. It is at its maximum mutation and it's proliferating way out of it. Now what happens is that we have diphosphate group right here, PIP2. It's a diphosphate group which is supposed to be converted to triphosphate group in order for it to get activated. P13K helps in converting diphosphate group to triphosphate group. What other structure helps us? What other ligand helps us, or what other system helps us in getting this desired effect? It's called PTEN. That's how you have to remember it. It's actually the name of it, PTEN. It acts on it and helps conversion of PIP2 to PIP3, diphosphate to triphosphate. But then this PIP3 is supposed to get converted back to PIP2. It is supposed to get converted to diphosphate group. But in case of mutation of PTEN, now PTEN, what does PTEN do? PTEN is actually a group of phosphatases. And what does it do? That it converts PIP3 back to PIP2. Otherwise, what are the effects? If there's loss of expression of PTEN, what are the effects? It's going to convert into AKT. What is AKT? It's actually protein kinases. Then these triphosphate groups will keep converting or keep producing the protein kinases. What protein kinases will do if they are mutated, with the help of PDK-P1 and PDPK2, what will happen? That mTOR will be produced. Now, what is mTOR? mTOR is actually a level of this genetic expression that it is at its maximum mutation and it is producing the most devastating effect at this level. So this will lead to cell growth, proliferation, and survival. Now, why are we saying that it's leading to cell growth, proliferation, and survival? Because those systems which were responsible for keeping a check on them have been deactivated. Let me show you which systems have been deactivated. RAS has been deactivated. RAS was supposed to keep it in check. Reticular activating system was supposed to keep it in check. If it's not activated or if it's not deactivated, there was a problem. Now it's activated all the time and cells are proliferating at the maximum capacity. RAS was supposed to keep a check on them, that how much the proliferation is going on, how long the proliferation is going on, when is the time for me to convert back to my inactivated form. Now if it's activated, what happens? All of these devastating effects will happen. Now if there's no check, cell growth will happen. No body is there to stop the cell growth from happening. Now proliferation, let's get to proliferation. How does proliferation is happening? How do proliferation is happening in this scenario? When PTEN loses its expression, there's no way of conversion of triphosphate to diphosphate, or it's what we can say into its less potent form or less active form. Now it's gone. Now the production of triphosphate will be at the maximum level. That will lead to the production of protein kinases, and that in turn will lead to devastating effect of proliferation and survival, because no structure in this whole system, in this whole receptor activation system, is taking the responsibility of controlling the whole cell proliferation and growth. So that's what you learn from this slide, that when these two mutations, P85 and P110, when those are mutated, they will lead to one way or another conversion of PIP2 to PIP3, and P13 does the part and PTEN does the part. When all of these are taking part and conversion of diphosphate to triphosphate, this will lead to cell growth, proliferation, and survival. And on the same hand, not on the other hand, on the same hand, if reticular activating system is activated all the time, cell proliferation will be at its maximum level until it reaches the mitogen level and then the extracellular kinase level. And that's how the cell growth, proliferation, and survival will go on, but there will be no check for it to stop it or to control it. I hope you get my point. Next on, we have signaling after receptor activation with the help of MAPKs. What does it do? It has this epidermal growth factor receptor. It's not estimated glomerular filtration rate, it's actually epidermal growth factor receptor, and you can see this receptor site on it, so it won't be confusing. So what does a receptor need? What does a receptor gets for itself to be activated, for itself to work? A ligand. And which ligand? This ligand. TG alpha will get attached to it. It's actually TG alpha which gets attached to it only. There's no other receptor, and it also has this EGF beta-cellulin and epiregulin. So what does it do? It activates SOS and GRB2. Now these are the pathways which will lead to the activation of RAS, reticular activating system. Now this reticular activating system seems normal here. Why does it seem normal here? Because we have an off and on switch on it. It's not written here that it's always going to be mutated. So in this system, in this receptor activation of MAPKs, RAS system is normal, reticular activating system is normal, and it has the off and on switch. It will be off at the level of GDP, it will be on at the level of GTP, and which of the structure was giving it the phosphate group? It was actually another GTP which was giving it the phosphate group and converting it into GTP, into its active form. After cell proliferation, normally it would go back and the cycle will keep happening in a loop. So in this diagram, in this topic, we're going to assume that now the RAS is normal. There's an off and on switch on it. So it will be converted into RAF. We talked about it. Further on, it will be converted into kinases and cytokines. It will include MEK1, 2, and afterwards ERK1, 2. These are kinases, cytokines. Now these will be activated and they in turn will get to the nucleus. Once they reach the nucleus, once this message has made its way up to the level of nucleus, what does it do? In the nucleus, it has 4 other things to attach to or to affect to. They include CJUN, CMYC, ETS, and CFOS. Now, these are not abbreviations, these are genes. These are genes present on your chromosomes. Now, it depends that which kind of message has been sent and which of these or many other will get affected because of it or will have the consequence on it. So, what happens afterwards when it gets the message? Cell proliferation, survival, and metastasis happen. Now, if there was off and on switch, you might be wondering that why it was normal. Yes, it was normal, but we're talking about the mutation. So if it gets mutated, what will happen? We talked about it will get converted into RAF. Then we'll have cytokines, we'll have kinases, which will go into the nucleus, which will act on the chromosomes, which will act on these four nucleotides and structures, and in the result, cellular proliferation will be happening and survival and metastasis will happen. Why cellular proliferation? Because of RAS. Why survival and metastasis? Because we talked about it. Because survival and metastasis happens if all those structures, all those organelles, all those places who were supposed to constrict them, who were supposed to keep a check on them, have been deactivated. So they will easily proliferate, survive, and then metastasize. Now, this whole system will in turn give more and more TGF alpha. More and more TGF alpha will come in, more and more TGF alpha will bind here, more and more this reaction will be happening, and this is actually a loop. This is a vicious cycle which once begins, it never ends. It will keep proliferating until it bursts that cell and come out of it and gets to another cell. Next now we're going to talk about phospholipase C. What's the phospholipase C pathway? So we're going to talk about a normal physiology here of a phospholipase C receptor. So this is the receptor of phospholipase C. It's attached to GQA. We talked about GQ pathway. We talked about G protein coupled receptor pathways. We talked about that there's going to be GS, which is synthesis. We talked about there's going to be GI, which is inhibitory, and then we talked about that there's going to be a GQ. Now GQ was supposed to increase the intracellular calcium, and it has another important function, and we're going to learn about it in this slide. So what happens? It affects on the PLC, and in turn the lithium will give off to lithium and diphosphate group will be in interaction. So PLC will give off its phosphate group and it will be converted into IP3. Now these have two intermediates, these have two resultants. Either it's going to be converted into IP3, either it's going to be converted into DHG. Now what does IP3 does and what does DHG does? Now that's where the function of this whole system lies. We talked about it gives off calcium. What was the other function we talked about? We didn't talk about it. We're going to talk about it right now. So what does a DHG does? It's going to give PKC, which are protein kinases. And what does protein kinases does? We talked about it. It increases those levels. It increases that pathogenicity. It increases that neoplasia activity. So more protein kinases will be secreted from DHG, will be produced from DHG, and for ester will be picking it up. For ester will recognize it, and neurotransmitter release and cell proliferation will happen. Now this PKC is not only affecting the regional level, it's also affecting the neurotransmitter level, and it's going to cause neurotransmitter release and cell proliferation. Cell proliferation was already happening, but there was no involvement of neurotransmitter up till now. So PKC took care of neurotransmitters and their release and their proliferation. Next, now we're going to talk about the functions of IP3. Now we talked about it's going to increase the calcium production. So yes, IP3 is converted into calcium, Ca plus 2. Now Ca plus 2 also has the same function. It also has neurotransmitter release and cell proliferation effect. How does it increase the cell proliferation effect? It gets inside the cell, it reaches up to that level that increases the calcium level of the cell and gets the cell up to that maximum potential for it to proliferate. And on the other hand, it is a perfect neurotransmitter. You might even feel it, that it's a very important even electrolyte inside the body. Even if hypocalcemia happens, there are many drastic effects on the body. So this cycle gives us PKC and calcium, both of whom have a very major role on neurotransmitter release and a cell proliferation effect. Next on, we have transforming growth factor beta. That's what we call TGF beta. Whenever we hear the name TGF beta, you have to go for transforming growth factor beta. Now we're going to talk about that tumor suppressive effects. It has tumor suppressive effects. And it causes apoptosis, differentiation, and maintaining the cell count. Next on, if there's failure to suppress the tumors, what's going to happen? Autocrine, mitogen, and motility. Now, first of all, that was a function of it. It causes the apoptosis. What apoptosis means? Programmed cell death. Programmed cell death. In what term we call programmed cell death? When we see that a cell is proliferating, or a cell has a foreign DNA or a foreign RNA or any kind of virulence, it is going through a cell cycle. So it's stopped at a checkpoint. It either can be G1, it either can be G2. At either of these checkpoints, it's stopped, and there's a whole trial that either we can repair that DNA or not. If we can't repair that DNA, what do? Caspases are induced inside the cell, and they kill the cell by lysing the cell and by lysing the organelles. That's what is called apoptosis. So tumor suppressive effects, it gives off tumor suppressive effects by apoptosis and differentiation of the cell and maintaining the cell count. That it should be appropriate cell count, it should not exceed that limit, it should not be below that limit. But what if it's a failure to suppress that tumor? It's autocrine, mitogens, and motility. It will cause the motility of the tumor that will lead to further devastating effects of invasion and metastasis. What's the difference between invasion and metastasis? Invasion is local. Whenever there's a tumor of breast, it invades inside the tissue and then goes to the lymph nodes and then to the axillary region. That's called invasion. It can invade to the bone level even. But what is metastasis? When it reaches, metastasis means when it reaches the homogeneous level, it involves the blood. When blood is involved, its cancerous cells, its tumor cells can migrate through blood to other organs, to other organelles, or to other places inside the body. That's what metastasis means. In a metastatic patient, you see there's a distant secondary origin of the tumor. That's what it's called. So it contains of myofibroblasts, malignant cells, and extravasation. Next on, we have metastasis. First of all, we had invasion and metastasis. Then it has reached to the maximum level, it has reached the final level, and that stage is metastasis. And it involves microenvironment change, osteoclast mobilization, microenvironment change. What does microenvironment change means? It means everything is being changed in that environment in which it's being metastasized. Microenvironment means that angiogenesis will be happening. For example, there's a tumor, it needs growth, it needs blood. What will happen? New blood vessels will form to support it, because there's nothing to stop it from now happening. So microenvironment changes will happen. Then osteoclast mobilization will be happening. There will be bone deformities, there will be bone weakening, shortening. All of that will be happening because of it. Next on, we have transforming growth factor beta, which we just talked about. And we're going to talk about how this affects granulocyte CSF. What does CSF stand for? Colony stimulating factors. Colony stimulating factors, not cerebrospinal fluid. It's colony stimulating factors. Now we're going to talk about which of these are tumor dependent and which of these are tumor independent. First of all, let's talk about tumor independent functions. All of those functions which are independent of the origin of the tumor, which are independent of the tumor. And those functions include tumor inhibitory functions and tumor stimulatory functions. These two functions are independent of this tumor independent function of transforming growth factor beta in GM-CSF. We talked about what GM-CSF is. It's actually granulocyte colony stimulating factor. So these are the tumor independent functions. Now let's talk about the dependent functions. Which of the functions depend on the tumor? And it contains CD4, CD8, T helper cells. Now all of these are reacting to the tumor. These are tumor dependent. That's why they're called tumor dependent structures. Now these include helper T cells, these include cytotoxic T cells. So all of that is happening at the same time. Then we have mast cells and natural killer cells. NK cells mean natural killer cells. Now mast cells are coming into the mix, and natural killer cells are coming in, rafting in to kill that foreign material. Next on we have macrophages. Macrophages will be rushing in, and that's how a granuloma is formed. When all of these structures are coming in together and they're unable to engulf the pathogen, they're unable to eat the pathogen, they're unable to defeat the pathogen, that's when granuloma is formed. Next on, we're going to talk about steroid hormones. Steroid hormones include progesterone and estrogen receptors. So these steroid hormones attach themselves to the progesterone and estrogen receptors and receptor for androgens and corticosteroids. Next on, receptors for vitamin A and D and fatty acids are also included in steroid hormones. Next on, we have membrane-bound mucins. What does a mucin mean? Mucin means a protective shelling. Mucin is actually a protective covering. Mostly it's present on the insides of our mucosa. It covers the epithelium, it covers the surfaces so that no harm could come to the epithelium or surfaces. So it covers them. But it also has a function. It also has a function of converting the message, of getting the message to the right place. So what happens when it gets in contact with ERB2, extracellular domain acts on it with the help of EGFR, with the help of these, it acts on it. And what are the functions of it? So mucins are actually of many types, but here we're going to talk about M1 and M4. Those are the most virulent types. Those are the most important types for us to understand in neoplasia. What do they do? What they do is that they increase beta-catenin production. They have very different effects in all of these. They increase the beta-catenin production. What does a beta-catenin does? It increases the cell differentiation, increases the cell proliferation. It decreases the P53. What does the P53 gene does? We talked about it in the last lecture of pathogenesis of neoplasia, that P53 is a tumor suppressor gene. First of all, it stops the cycle at a certain point, at the same point where it's not supposed to be going in the wrong direction. It stops the cycle. On the second place, it tries to make that DNA normal again, make that DNA work again. And in the case it doesn't, what does P53 does? It induces the caspases which are programmed for apoptosis. It causes that cell's death. So if its production is decreased or if it's mutated in some way, now P53 is out of the way. Now we have ER, which are estrogen receptors. It increases the estrogen receptor proliferation. Now estrogen receptor proliferation will increase, the cells increase. So it has very different effects on all of these. What does it want? It actually wants the cells to proliferate. It's actually neoplasic in nature. These M1 and M4 cells among the mucins have very neoplasic effect. So M1 and M4 will lead to all of this, and this will lead to formation of neoplasia. So you can see this is the cytoplasmic tail of the extracellular domain. Extracellular domain is also called molecular domain. Keep that in mind. Next on we have transcriptional activation. How does it get activated with the help of transcription? We have Fos and Jun which act on AP1. Now AP1 is a protein. It's a very important protein which is related to these Fos and Jun. What happens is AP1 protein gets activated, then it binds to the DNA. It binds itself to the DNA at a very specific certain point. It's not like it can bind anywhere inside the whole DNA. It has a very specific place. It binds to that place, and what happens afterwards? That proliferation will happen, invasion and metastasis will happen, angiogenesis will be happening, and survival will be happening. Now, some terms that you don't know, let me give you a detail on that. So first of all, when AP1 came in, that's a protein. And what does AP1 do? It controls all of these functions. It controls the proliferation, it controls the metastasis of the cell, it's actually tumor suppressor in its nature. Then we have angiogenesis. It starts even the angiogenesis. What does angiogenesis mean? Angio means blood vessel and genesis means production. It means blood vessel production to those areas which are hypoxic in nature. Whenever the tumor cells will form, what will happen? Maybe it won't have enough blood supply. So what will happen? Angiogenesis will be happening. We're going to talk about angiogenesis in detail, that how does it happen. Then when all of these things are happening, when they're getting enough blood, when they're getting enough nourishment, when they're invading and metastasizing at the same time and proliferating, survival will be happening because all of the immune systems are down. All of those systems which were supposed to take it down are over. They're not working at the capacity. They're either mutated or turned down. So that's how the cells will be surviving. Next on we have androgens and estrogen receptors. We talked about androgens and estrogen receptors. We talked about what androgen and estrogen will be binding to. They will be binding to androgens and estrogen receptors which are present on the nucleus. Mostly. In some cases maybe cytoplasm, but mostly it gets to the nucleus. So this is, for example, a hepatocyte. So what happens that this is the nucleus, it reaches its level, it gets to the nucleus, and then what happens? This is androgen, this is estrogen. Now androgen gets attached to a specific part of chromosome, estrogen gets attached to a specific part of chromosome. Now both of these ligands have attached themselves to a very specific part of the chromosome in order for them to get a specific messenger RNA. Now when the messenger RNA is produced, proteins will be produced according to their need, according to what the information they got in. So we talked about it. Out of these androgens and estrogens binding to the chromosome with their ligands, messenger RNA will be produced. That messenger RNA will lead to the formation of protein. A lot of amino acids will be forming in, and the amino acids will combine together and a protein will be formed. Now we have a very difficult term, cellular senescence. You might be wondering what a cellular senescence is. Cellular senescence means that a cell has been halted. A cell has been halted in its progress. It's not doing anything now. Now, cellular senescence is a double-edged sword. It can either stop the proliferation of cancer at the stage it is at. It's not going to let it move forward with the stage. It's not going to let it replicate. So yeah, we have multiple beneficiaries of cellular senescence. But what are the side effects? With the help of other chemicals and substances, it can actually increases the proliferation. So we might never know. But the term cellular senescence should give you an image in your head that the whole process is stopped. That's called cellular senescence. So DNA damage response, if cellular senescence is happening, what is going to happen? It's halted. So if the DNA is the problem, if there's mutation in the DNA, we're going to talk about DNA damage response. DNA damage response will be happening, that we talked about in the last, or the, you can say, first lecture of carcinogenesis, that how does the response of DNA damage comes through. DNA damage response comes through, it helps the DNA to get back to its shape, and that's how cellular senescence will end. That's how this will get back to normal cellular stage. Tumor suppressor activity will be seen, that how a tumor is being suppressed, if it is being suppressed or not. Next on we have oxidative stress, ROS it's called. Oxidative stress will be monitored in this case, that if there's any kind of oxidative stress, we have to remove that. Next on we have cytokines like IL-6, IL-8. Now these cytokines are released in response to this kind of antigen, on response to this kind of response from the body. So that gives you a glimpse of cellular senescence. If you want to learn more about it, you can find it on Wikipedia's. But one thing as a medical student, you have to know that it's a halting process. Next on we have telomere stability and tumor-genesis. Now we all know what telomere is. When a chromosome, which has two alleles, different alleles, when its smallest part is cut down, it's called telomere. So what happens is that progressive telomere shortening will be happening. You can see now these ends are gone. Those telomere are shortened. So what does a shortened telomere does actually? It's going to get to the fusion stage. Now these both ends will fuse. You might be wondering why they are fusing now. First of all, what happened? Telomere shortening. Then fusion has happened. Why are they fusing? Because they have to get to the stage of division and the chromatids will be separated apart. It's just like you had these two fingers and they were just like this, and by the telomere shortening they got into this shape, and now they have to get into zigzag shape in which they will be cut down equally for both cells. What happens, there will be a bridge formation between these two, and it's supposed to be cut down right here in the middle so that both of them get equal amount of chromatin, equal amount of DNA material. But it doesn't happen so, because we're studying the neoplasias, so it won't be happening. What will be happening is that in anaphase, either one part will be subjected to deletion, either one of them will be subjected to amplification. We talked about deletion, that if one segment of a chromatin material, one segment of the chromosome is cut down, it's deleted, then it's called deletion. And amplification means if one part or one nucleotide or very minute part of a DNA is replicated more than one time, it's called amplification. Now they are not, both of them are not equals. So what happens is that this kind of formation will be happening. It's not even. So non-homologous chromosome will be seen, and breakage, fusion, bridge will be formed. Breakage, first of all, this breakage happened, then fusion happened, then again breakage happened. So that's why it's called breakage, fusion, bridge. Next, I'm going to talk about it a little more briefly, that for example, this was a chromosome and this was its telomere. What happens is that first of all, with the progressive mitosis, telomere shortening will be happening. How is that going to happen? You can see it's being cut down right here. So telomere shortening happened. You know the next process, what will happen afterwards. This shortened telomere is now here, and this whole chromosome or this whole body of it activates p53 and RB. Why are they activating the p53 and RB at the same time? Because they are detecting a mutation. We talked about in a normal cell, whenever a cell goes through mutation, whenever there's a very minute mutation in the cell, what happens? The cell cycle gets arrested at the level of G1 or G2. It can be either one of them. But in most of the cases, it's going to be G1. So if at the level of G1, it's arrested, so p53 will be activated. Now p53 is responsible that if it can take care of this telomere or not, if it can repair it or not. And if not, what will happen? Then RB will be activated. What will RB be doing? RB produces all of those cytokines which are required for the cell cycle to continue. So we talked about it can either be checked at G1 stage or at G2 stage. G1 by S and G2 by M, these are the checkpoints of the cellular cycle. Gap 1, G1 means gap 1, S means synthesis phase, G2 means gap 2 and M means mitotic phase. So it can either be checked at any level, and if there's any kind of pathology seen, all of these will be activated and they will be halted at these stations. And replicative senescence will be happening. We talked about senescence just about now. What does senescence mean? A halt in progress, a stop in progress. If a cell is at second stage of cancer, it won't move to the third stage of cancer. It won't mutate afterwards. It will go into senescence. So what does a replicative senescence means? It means it's not replicating anymore. It has been sent into a coma. So it's not replicating anymore. Now we have time to take care of it. And this P53 will try its best to get this telomere straightened, to get this telomere into normal shape. But if it doesn't get it, what does it do? It induces those caspases we talked about. It induces the apoptosis, and this will lead to cell death. Afterwards, we have oncogen-induced cellular senescence. We talked about normal senescence. We talked about replicative senescence. Now we're going to talk about oncogen-induced cellular senescence. It means what are those oncogens and how do they induce cellular senescence that they should stay at that state. They should not move afterwards. Oncogen activation. How does an oncogen get activated? We talked about it in the first lecture of carcinogenesis. There's normally a proto-oncogene which under different conditions, it can be either any chemical or any carcinogen or UV light or whatever, it gets converted into oncogene. Now oncogene, where proto-oncogenes were responsible for normal growth patterns, normal cellular growth, oncogene won't be holding all of that role. It's going to be unregulated now. So oncogene activation will be happening because of this DNA damage, and more of the DNA damage will happen because oncogene is activated, and then oxidants will come in. They'll also act on it. So DNA is damaged, which will lead to DDR, which is ATM, ATR, CHK1, CH2, all of these cytokines will be activated. And this in turn induces interleukin. IL-6 means interleukin 6 and interleukin 8. Now they'll press upon their functions, and this oncogen activation will also activate it, will also have effect on interleukins and 6 and 8 and SAS. So all of this because gene is being damaged, because there's mutation, P53 will be activated at the same time. P53 gets the gist of it that, okay, mutation is happening. I have to get activated. I have to halt the process. If I don't halt the process, carcinogenesis will start. So P53 is activated to take care of this mutation. And this P53 will put it into senescence so that it can get the time to either repair this damage or induces those caspases to get the apoptotic effect. This oncogen activation also induces P16 INCA4A, which is very important for this whole process, and it involves RB. When RB gene is activated, all of those cytokines are under the thumb of it. Now those cytokines are not being produced. Now when those cytokines are not being produced, what's happening? Cell cycle won't continue, because cell cycle cannot continue without the help of cytokines. And this will lead to SAHF, and in turn it will also lead to senescence. So in short, if the body recognizes it, if the cell's mechanisms are on, they'll send this kind of cell or a mutated cell into the senescence. They won't let it replicate. And if they're unable to solve the mystery, if they're unable to make it right, they cause the apoptosis. Next, now we have role of telomere attrition. What is role of telomere attrition? Now, this includes colorectal carcinoma. So you have to be very thorough about it, that how does it develop. Now, this is a normal colon. Normal colon working at its best capacity, doing its normal functions. When telomere attrition happens, meaning telomere destruction happens, what happens? And telomere attrition doesn't happen overnight. It happens because of repeated cell cycling. So what happens? Chromosome ends are uncapped now. When the chromosome ends are uncapped, what will happen? chromosome instability will be happening. Now, this colorectal carcinoma has actually two pathways, so from which it can get to colorectal carcinoma. But the one we are discussing here is because of chromosomal instability. So chromosomal instability develops because there's any kind of attrition. When there's telomere attrition and the chromosomes are uncapped, and when they're uncapped, when there's no production, when there's this chromosomes instability, what will be happening? This kind of result will be seen. You can see this is a normal colon, but you can see now more and more of those crests and more cellularity present inside of them. So this is adenomatous polyp. A polyp has been produced because of chromosomal instability. And when chromosomal instability leads to adenomatous polyp, it's because of aneuploidy mutations. These mutations include one part. That's why it's called aneuploidy. So this mutation will lead to the formation of adenomatous polyp. And survival of malignant cells via telomerase activity will be starting from right here. But how does it reach to this level of colorectal carcinoma, invasive colorectal carcinoma? Because those chromosomes which were responsible for its normal functioning has been uncapped. Attrition is happening, they have been uncapped, and they are unstable. And when they're unstable, polyps are developing. And when the polyps are developing and nobody is taking care of them and there's chromosomal instability, this will in turn lead to invasive colon carcinoma. Next on we have programmed cell death. What do we call it? Apoptosis. We talked about it multiple times in the terminologies. So apoptosis, this is what a depiction of apoptosis will look like. You killed it just by programming it that okay, we are inducing caspases in it, it should die in right about this time. That's called programmed cell death or apoptosis. Now that we have seen this depiction, now let's talk about apoptosis. P53 and TP53 genes are very important for causing the apoptosis. We talked about that if there's any kind of mutation present inside the cell, if there's any kind of deformity present inside the chromatin of the cell, TP53, P53 will be activated. Once they're activated, what do? They put the cell into senescence, stop and halt the process of the production. And once the production and this process is halted, what does happen afterwards? Afterwards, P53 will start doing its functions. What are the functions? First, it will try that I should make this sequence right and put it back into the cell cycle, but won't happen. In most of the cases, it won't happen. So what's the second option for it? It activates all the systems, plus it induces those caspases and causes the apoptosis. Next on, in follicular lymphoma, apoptosis is seen because follicles are being involved and it's lymphoma, it's affecting the lymph nodes. Apoptosis is being induced in those cells too. Now, anoiasis, or we can call anoiasis, is form of apoptosis. What does that mean? What is anoiasis? Anoiasis is a term used when a cell loses its attachment or its contact with ECM, which is extracellular matrix. When it loses its contact with extracellular matrix and it goes away and it loses its contact with the neighboring cells, that's called anoiasis. It's a form of apoptosis. It's a form of programmed cell death that we're letting go of that cell. It won't get any nourishment. It will just die off within this time. So this is a programmed cell death, and this is the type of apoptosis. Next on, we have oncogene mediated apoptosis, in which oncogene decides at which point this cell should die. This cell should go to senescence, and then it should die. So that's also one type of apoptosis. Next on, we have angiogenesis. Now, what is angiogenesis? I talked about it in the earlier slide. Angio mean blood vessels or blood related structures, and genesis mean production. It means production of blood vessels. Why the production of blood vessels is happening now? So let me give you a brief overview of angiogenesis. Angiogenesis happens when a tumor cell is growing at a place where there's no blood supply or very minute blood supply, and it needs the increased blood supply. It affects the tumor suppressing factors, and afterwards what happens is that any vessel which was going right past through, for example, this is a tumor and this is a vessel going through. First of all, this vessel will perforate because it's right here, so it will perforate and get the supply right here, give the supply to the cancer cells, and in turn they'll proliferate, they'll grow. So let's learn it step by step so that you can understand in a better way. First of all, what happens is that proteolytic enzymes will come in. Why are proteolytic enzymes coming in? This vessel, this is the cancer. This vessel needs to be broken down. In order for blood to come in, first we have to destroy this wall of the vessels. For that, proteolytic enzymes come in and they perforate post-capillary venule of the basement membrane. BM stands for basement membrane. So once they perforate this basement membrane of the post-capillary venules, they reach the tumor. Now what happens? Now endothelial cells will proliferate and migrate towards angiogenic cytokine source. Now the endothelial cells will come in to form the new vessels and they will migrate towards that side where it's being formed. Next time we have lumen capillaries. Now the small lumen capillaries will be forming at that site. They're immature capillaries and they are invested with basement membrane. Last but not the least, tip cells will be formed. Now these are joining of similar cells to produce that network. Now all of those cells are being, for example, if it was a very small capillary in size and it was not that big to provide the cancer cells with enough nutrition, now the same cells are coming in and binding, and this is getting stronger and stronger, and the blood supply is getting better and better for the cancer cells to grow. That's what angiogenesis means. So most of the cells go through, most of the cancers make a person go through angiogenesis. Mediators of tumor angiogenesis, which is called VEGF. What is the mechanism of it? Let me tell you that tumor cells, for example, have genetic factors which have more oncogenes, less tumor suppressive activity. What does that mean? That genetic factors are there in the tumor cells, there are more oncogenes but less tumor suppressive activity because the tumor cells, these are not actively working, they are just tumor cells. They have less tumor suppressive activity because they're not acting in that way, and they have more oncogenes. More oncogenes mean more proliferation, more unregulated proliferation of the cell. On the other hand, extra tumor cells have this hypoxia, chemokines, cytokines, growth factors, and mediators, all of these. Now, all of these are responses to tumors actually. This hypoxia, this chemokine, this cytokine, this growth factors, and these mediators are actually response. What happens is that they activate VEGFs. What happens afterwards that they bind and activate these receptors. Now this goes on, VEGFs go on and activate their receptor. Once the receptors are activated, what does happen? Now, before we go on to it, there are three kinds of receptors, right? VEGFR1, VEGFR2, and VEGFR3. So there are three kinds of receptor present. So who decides what to bind and where to bind? First of all, it's going to attach itself to beta, VEGF beta. And on the other hand, PIGF will also be attached to the VEGFR1. Now these terminologies might look difficult to you, but these are very important and you have to understand them one way or another. Afterwards, VEGF-As get attached to both of these. One side, VEGF-B is being attached to one side, on the other hand, VEGF-As are being attached to both sides of the chains. And then comes VEGF-C. So we have basically these A, P, and C, and then we have this PIGF. On the other hand, D attaches right again to both of these, VEGFR2 and VEGFR3. What happens afterward? What are the side effects? Now that's what's important. That was just VEGF binding to its receptor. we learned there are 4 types: A, B, C, D. Among them, we learned some bind 2, some bind 1. What are the side effects? What are the effects of it? Effects are vascular permeability increased. It will be more and more vascular permeability for that area. Proliferation will be happening because of these cells, because of these PLCs, because of RAF, because of MAPKs. Proliferation will be happening. Survival will increase because all of those factors which prohibit the formation and proliferation of these cancers are gone now, or either inhibited or either mutated. So survival will be happening. Next on, the migration will start happening. Once they have survived, once they have gathered in one place, what will they do? They'll move from that place. They'll go on, they'll involve the blood vessels and they'll go on to infect the other area. So they migrate and then they mobilize. First, they migrate into the blood vessels, then they mobilize and go into the other organs and affect them in return. Next now we have tumor invasion and metastasis. In situ means in the very form, tumor at primary site leads to invasive tumor at the primary site. First of all, there's a tumor where in situ, tumor in situ means the tumor where it was born, where it was produced, it's present here. It firstly gets converted into invasive tumor at the primary site. First, it invades that area. For example, if the tumor was at breast, and there was a little lump, first of all, it's going to invade that area. Then the tumors will be within the vessels. Now it will go deep and it keeps invading until it reaches the blood level. And when it reaches the blood level, what's going to happen? It's going to be within the vessels. And when it's within the vessels, entry into the distal organ site will be very much possible. It will get inside that blood. It will go on to any organ it wants. And basically it goes on to those organs which are very rich in blood supply, like liver, kidneys, heart, lungs, all of those areas which are very rich in blood supply. And then they go into a dormancy phase. They're dormant for some time because they have the whole infectivity thing, the whole cycle will start again. They'll go dormant. And when the dormancy phase is over, the growth start back. The growth starts back. So first of all, they were at a place, then they invaded that place, then they got into the bloodstream, then they migrated to the distant organs, they went dormant, and at the end, they grew. They went for the growth. Now, tumors which went within the vessels can go back, can recede back into invasive tumor at the primary site. This happens all the time with the modalities, with the treatments, and in some cases without even one of them. So that has been shown through researches that it can go back, it can recede back. On the other hand, if there's a tumor in situ tumor at primary site, it can directly go and get into the other organ without going through any of these effects, without invading the tumor, without getting into the vessels. If the organ is nearby, it can actually get to that organ without even one of them, and with the help of soluble mediators, of course. With the help of these soluble mediators, they prepare the distant sites and they get that entry into the organ site without going into these modalities, without going into these phases of metastasis. On the other hand, we see this dormancy. This dormancy can recede back and see invasive tumor at the primary site. This dormancy can stay there and it can recede back to invasive tumor at the primary site. Recede back means go back a stage, go back a place. Next on, we have tumor invasion and metastasis. Now, carcinoma in situ cell becomes capable of invasion. Obviously, when a cell is there and it's in situ position, the carcinoma has now become capable of invasion. When does the carcinoma become capable of invasion? When it has achieved its growth, which was required for it. Then it's capable of invasion. What happens is that this is the basement membrane. For example, it's a basement membrane. What happens is that, okay, the tumor is right here. Now it wants to invade. So what happens? Tumor cells undergo EMT, traverse basement membrane and invade through extracellular matrix. Now tumor cells undergo EMT. They go through this whole shrinking process. They traverse basal membrane, they affect the basal membrane and invade through extracellular matrix. So now this has opened up. Now it's releasing all of those DNAs and its products which are present inside the cell, and they go inside the cell without any problem. So this releases the proteolytic enzymes. Those proteolytic enzymes break off this bondage, and next on what happens is that it can either get into the blood vessels, it can either get into the lymphatic system. Once the cell has burst, it has released its proteolysis, and now it has breached that basal membrane and now come out, and now it can go either into blood vessels or either into lymphatics. Now in blood vessels, it can go through intravasation, meaning through the blood vessels, intravasation, and they can survive in blood. There's a whole better survival rate in the blood. And from the blood, they can go to any organ. They can go to the lungs, they can go to the brain, they can metastasize and go any place they want to here. Now it can also extravasate and lead to the desired effects or the effects that cancer causes equally like it does in the intravasation. Now we have epithelial mesenchymal transition. Now this is an epithelial layer. Now how does an epithelial layer convert into a mesenchymal layer? That's what this topic is about, epithelial mesenchymal transition. So what happens is that one of them loses the cell adhesion power. They adhere to each other like this. They're very tightly bound. One of them will lose that adhesion power. Now it's an empty thing right between all of those adherent structures. It's not fitting in. So what will happen? It will try to shrink out. These adherents will force it out. These adherents will try to force it out. So what happens? This gets out of it, and now it becomes a part of mesenchyme. So that's how an epithelial mesenchymal transition goes through, that one cell loses the adhesion, is shrunk, and it's forced out of it into mesenchyme, and then it becomes a mesenchymal cell. That's how this epithelial mesenchymal transition goes through. As you can see, this constriction happened, and then remaining epithelial layer is still that way. They adhered back to each other, but this cell went away, and now it's a mesenchymal cell. Next, now we have cancer cell ecosystem. What does a cancer cell ecosystem means? Meaning, in what system cancer cell propagates, or in what system cancer cell has around it. What are the things that, if you look at a cancer, what will be looking at, what structures will be following it, or what structures will be surrounding it? If this is a cancer, what structures are around it? What structures are surrounding it like a globe? First of all, macrophages. Now, macrophages, whenever there's any kind of mutation or infection or cancer, macrophages rush in to do their job, but they can't engulf the cancer. They can't do anything about it. So they just keep getting there. They just keep accumulating there. So that's what you see. It involves neural cells. If the involvement of neural cells is involved, it involves the fibroblasts. It involves the adipocytes if liver is involved. It involves the endothelial cells if the basal membranes are involved. It involves the leukocytes. All of these structures are combined together. They're just a matrix. They're just a mix of it. And that's what the ecosystem of tumor is. Next now we have tumor cell stromal interactions. Now look at this slide. This is a stromal growth factor. This is a stromal growth factor. Now these are chemokines, inflammatory cytokines, immunomodulatory factors that we just talked about. That in a cancer we'll see these chemokines, inflammatory processes, and these stromal growth factors. Now what will happen? That this will secrete, these fibroblasts will secrete angiogenic factors and proteases to try to catalyze it, but it won't. So this will become a part of it. On the other hand, these chemokines, inflammatory cytokines, and mediators will secrete angiogenic factors and proteases. Now these angiogenic factors will provide the blood supply, will provide it with the blood supply that is coming from downside, and proteases, just like it, but it won't have any function on it. Next on we have adipocytes, which will secrete leptin, which in a normal person calls satiety, but it won't have any function here. It won't help in removal of the cancer, but it will help the cancer in becoming what it is afterwards. It gives it interleukin 6. And next on, we have these growth factors and growth factor invasion factors. All of these will be combined together in it and make that matrix. Next on, we have tumor cell arrest. What does a tumor cell arrest means? Tumor cell arrest means that there is tethering. This is tethering. There are either E-selectin or P-selectin. We're going to talk about it. Now, this is tethering, but this is stable adhesion. So what happens between these two is that the fluid is flowing in the blood vessels. So what happens? There's a tumor cell right around here. So what a tumor cell will do if it's floating inside that capillary, inside that blood vessel, and these receptors are present, what will it do? It will get tethered here. Now these receptors will get tethered here. Now this is called CD44. Now these receptors are tethered here, but tethered to P-selectin or E-selectin, not to a stable adhesion. They're either P-selectin or E-selectin, and in most cases you have to remember it's going to be P-selectin. That is just mentioned here. So P-selectin or E-selectin, but with time it develops that fibrin particle which gives it a stable adhesion. And right with stable adhesion, this fibrin factor, we have one Willebrand factor. Now, how does this happen? How in the hell that these came out? When the tethering happened and P-selectin and E-selectin took it for tethering, they gave off the message and they attracted the fibrin and the von Willebrand factors. They came in, they attached to the skin, they gave a stable adhesion, they got the CD44 cell and won't let it move out. So this is tumor cell arrest. They stop the cell before it reaches any basic organ, any organ which has higher blood supply, and it has kind of stopped a very important cell which would have caused a secondary tumor, or it would have led to metastasis. But now this cell has been arrested, so it won't be doing anything. It won't be causing any kind of secondary tumors in any kind of organ. So that's what tumor cell arrest is. Now these are integrins, you have to remember, and these are platelets. Platelets are present right here so that this tethering and this P-selectin and E-selectin can work on. And this is vascular endothelium. You all know this is vascular endothelium, and those platelets came on. They played for a bed, and then E-selectin and P-selectin got them tethered. They attracted the von Willebrand factors and fibrins. They came in, they made this stable adhesion, and this is called tumor cell arrest. Next on, as you can see, these are CD44 cells. We just talked about them, these CD44 cells. Now they are attached to either one of these receptors. Now this is a tumor cell membrane. Now these CD44 cells will get attached to this leukocyte or phagocyte. So L-selectin will be the one to whom CD44 is attached. An adherent leukocyte which is present right here, this adherent leukocyte is attracting this. But attraction is from both sides. P-selectin and E-selectin are also attracting it. But this L-selectin came in, which will adhere to CD44. Now let me explain it in a better way. We talked about it, this CD44 was free. This receptor was free. This receptor was being bound to P-selectin or E-selectin. Now, you should know that it's not going to be free all the time. It's actually getting to a stage where this adherent leukocyte will make a bond with CD44, and then P-selectin and E-selectin will attract these ligands so that these tumor cells are capsulated here or captured here or arrested here so that it won't move out of here. So these endothelial cell membranes will provide the surface for this whole reaction. Now these micrometastases. What does micrometastasis mean? Meaning very minute amount of metastasis. So if they're very micro, if they're very minute, what's going to be the fate of them? For example, this is a primary tumor, and with therapy, we cause the tumor death. Or this primary tumor may lead to gross metastasis. That minute micrometastasis has led to very gross metastasis. That cell has not been taken care of. That cell was not even found. That cell was not even diagnosed. It's moving from here. That metastasis was not even diagnosed. So it led to gross metastasis. Or this primary tumor may lead to micrometastasis. So this was a primary tumor. It had these three fates in the very start. Either it could lead to very early diagnosis. Diagnosis was very early. It was in first stage or in very early second stage. We gave it the treatment, we gave it the chemotherapy, we gave it the radiotherapy, and we got the tumor cured and the tumor death happened. Now the second option, that macrometastasis happened. Now the macrometastasis mean a bunch of cells broke off and went off to an organ, a very basic organ like lungs or liver or the kidneys. So it will lead to them, or it may lead to the bones, meaning it will go wide, it will go body wide. It can go any place. Once it's broken, it can go any place. Any cell can go any place. That's called gross metastasis. Now comes the micrometastasis. These are very minute metastases, meaning a bunch of cells or a cell. Now, what's their fate? The growth arrest happens, and if this growth arrest continues, that's what we call quiescence, right? Now, quiescence has happened. The growth has been halted. The growth has been arrested because they don't have that capability to divide themselves or to produce another tumor there. So with time, factor activating entry into the cell cycle, if they enter into the cell cycle, what will happen? It will lead to gross metastasis, because it's only possible if it's polyclonal, that any of the 20 or 30 or 50 or 1,000 cells will lead to cancer. But in the form of monoclonal, it only needs one cell. It only needs one cancerous cell. So only one cell may lead to all of these results. So even the metastasis was very micro, very minute, but it was just a cancer cell. It was just a cancer cell. It had its mutation. So with time, after going through this quiescence, it will go to gross metastasis. It will get all of those things needed, and it will replicate, and it will lead to gross metastasis. Now those primary tumors can also lead to this effect, in which activation of cell cycle can happen and lead to excessive cell proliferation. Now that primary tumor is not going through excessive cell proliferation. It's not stopping. Even that tumor is growing in size, let alone these all. These are further types of it. Now this excess of cell proliferation may lead to this kind of gross metastasis. So what we learned is that there was a primary tumor. If it was diagnosed early, it was treated. If it was not diagnosed early, it's going to metastasize and go to the distant place, and gross metastasization happened. Okay, if it was micrometastasization, what happened? It went through quiescence, it increased in size, it replicated, and then it's gross metastasis now. But if the primary tumor is increasing in size and cell cycle has been activated there, now that will lead to excessive cell proliferation, and that will lead to gross metastasis. That primary tumor will lead to gross metastasis. So what we learned in this lecture is about the pathogenesis of neoplasia, the pathogenesis of cancers, that how some are benign, how some are malignant, how we take care of benign, how we take care of malignant. Now, what does that malignant contains for it? What are the types of the malignant tumors? How do we define them? How do we get to know them? What are their histological features? And then we got to know about their ligands, their substances. We got to know about which ligands are required for which kind of receptors. We learned a lot about receptors, that what tyrosine kinase pathway is, what G protein coupled pathway is, what extracellular receptors are, and how their pathways work, and how the nuclear receptors work, and which of the ligands come inside the nucleus and do their function. We learned about quiescence. We learned about senescence, in which it's halt of the whole process. We learned about if there's a primary tumor, whether it can metastasize or not, whether it metastasizes, how can we deal with it if it's early diagnosed. And then we learned a lot about specific terms which are required to get to know the proliferations of the cells. How does it proliferate? How does it reach the next organ? How does it metastasize? So all of these information will be very handy if you go through this lecture and review both of these lectures together. Thank you for watching sqadia.com.